A DNA is one of the possible double helical structures which DNA can adopt. A DNA is thought to be one of three biologically active double helical structures along with bDNA and zDNA. It is a right-handed double helix fairly similar to the more common bDNA form, but with a shorter, more compact helical structure whose base pairs are not perpendicular to the helix axis as in bDNA. It was discovered by Rosalind Franklin, who also named the A and B forms. She showed that DNA is driven into the A form when under dehydrating conditions. Such conditions are commonly used to form crystals, and many DNA crystal structures are in the A form. The same helical conformation occurs in double-stranded RNAs and in DNA-RNA hybrid double helices. Structure: like the more common bDNA, a DNA is a right-handed double helix with major and minor grooves. However, as shown in the comparison table below, there is a slight increase in the number of base pairs BP per turn. This results in a smaller twist angle and smaller rise per base pair, so that a DNA is 20 to 25% shorter than bDNA. The major groove of a DNA is deep and narrow, while the minor groove is wide and shallow. A DNA is broader and more compressed along its axis than bDNA. The identifiable characteristic of a DNA X-ray crystallography is the hole in the center. A DNA has a C3 endopucker, which refers to the C3 carbon in the furanose ring being below the sugar plane. Comparison geometries of the most common DNA forms. Geometry attribute: A form B form Z form. Helix sense right-handed left-handed. Repeating unit 1 BP to BP. Rotation: BP 30 to 0.7 34.362. Mean BP turn 11 10 12. Inclination of BP to axis 19 1.29. Rise BP along axis to 0.6 y 0.26 nanometers 3.4 y 0.3 for nanometers 3.7 y 0.37 nanometers. Rise turn of helix 28.6 y to 0.86 nanometers 35.7 y 3.57 nanometers 45.6 y 4.56 nanometers. Mean propeller twist plus 18 plus 16 0. Glycosyl angle anti-pyrimidine, anti-purine, syn. Nucleotide phosphate to phosphate distance 5.9 y 7.0 yc, 5.7 y. G, 6.1 y. Sugar pucker C3, endo C2 endo C, C2 endo. G, C3 endo. Diameter 23 y to 0.3 nanometers 20 y to 0.0 nanometers 18 y 1.8 nanometers. B intermediates. Research also indicates that a form DNA can hybridize with the more common bDNA. These B intermediate forms adopt the sugar pucker properties or the base conformation of both DNA forms. In one study, the characteristic C3 endopucker is found on the first three sugars of the DNA strand, while the last three sugars have a C2 endopucker, like bDNA. These intermediates can form in aqueous solutions when the cytosine bases are methylated or brominated, altering the conformation. Alternatively, guanine and cytosine-rich fragments have been shown to be easily converted from B to A form in aqueous solutions. Biological function. A DNA can be derived from a few processes, including dehydration and protein binding. Dehydration of DNA drives it into the A form, which has been shown to protect DNA under conditions such as the extreme desiccation of bacteria. Protein binding can also strip solvent off of DNA and convert it to the A form, as revealed by the structure of several hyperthermophilic archaeoviruses. These viruses include rod-shaped rotaviruses served to SRV1, enveloped filamentous lipothrixviruses AFV1, SFV1 and SIFV, Tristromavirus PFV2 as well as icosahedral portglobovirus SPV1. A form DNA is believed to be one of the adaptations of hyperthermophilic archaeoviruses to harsh environmental conditions in which these viruses thrive. It has been proposed that the motors that package double-stranded DNA in bacteriophages exploit the fact that a DNA is shorter than bDNA, and that conformational changes in the DNA itself are the source of the large forces generated by these motors. Experimental evidence for a DNA as an intermediate in viral biomotor packing comes from double difarster resonance energy transfer measurements showing that bDNA is shortened by 24% in a stalled crunch to form intermediate. In this model, ATP hydrolysis is used to drive protein conformational changes that alternatively dehydrate and rehydrate the DNA. And the DNA shortening, lengthening cycle is coupled to a protein DNA grip release cycle to generate the forward motion that moves DNA into the capsid.